Ashwagandha is among the top medicinal plants. A number of scientific studies have pharmacologically validated the therapeutic potentials of ashwagandha. Immune boosting. A scientific study showed that root powder extract of ashwagandha was found to stimulate immunological activity in Babel C. mice. The study revealed enhancement in the total white blood cells count, increase in the bone marrow cellularity and alpha-esterase positive cell number, enhancement in the circulating antibody titer, and the number of plaques-forming cells in the spleen. Inhibition of the delayed type hypersensitivity reaction, enhancement in phagocytic activity of peritoneal macrophages. These results show the immunomodulatory activity of ashwagandha extract, which is known as a strong immunomodulator in traditional medicine. Ashwagandha as antioxidant. Researchers have discovered that some of the chemicals are powerful antioxidants. Studies conducted on rats' brains showed the herb produced an increase in the levels of three natural antioxidants, superoxide dismutase, catalase, and glutathione peroxidase. These findings are consistent with the therapeutic use of ashwagandha as an Ayurvedic anti-cancer activity. Ashwagandha is reported to have anti-cancer effects. Research on animal cell cultures has shown that the herb decreases the levels of the nuclear factor kappa B, suppresses the intercellular tumor necrosis factor, and potentiates apoptotic signaling in cancerous cell lines. One of the most exciting of the possible uses of ashwagandha is its capacity to fight cancers by reducing tumor size. In another study, the herb was evaluated for its anti-tumor effect in urethane-induced lung tumors in adult male mice. Following administration of ashwagandha over a period of 7 months, the histological appearance of the lungs of animals which received the herb was similar to those observed in the lungs of control animals. Effects against inflammation. Researchers explored the capacity of ashwagandha to ease the symptoms of arthritis and other inflammatory conditions. Its naturally occurring steroidal content is much higher than that of hydrocortisone, which is a commonly prescribed anti-inflammatory. Also, the effectiveness of ashwagandha in a variety of rheumatologic conditions may be due to its anti-inflammatory properties. Finally, ashwagandha doses to rates produced anti-inflammatory responses comparable to that of hydrocortisone sodium succinate. Heart protection. Researchers have evaluated ashwagandha in clinical studies with human subjects for its diuretic, hypoglycemic, and hypercholesterolemic effects. Studies on human subjects have shown that the powder extract of ashwagandha decreases blood glucose. Also, it significantly increases urine sodium, urine volume, and decreases serum cholesterol, triglycerides, and low-density lipoproteins. Effects against stress. Its anti-stressor properties have been investigated in a study using adult Wistar strain albino rats and cold water swimming stress test. The results indicate that the drug-treated animals show better stress tolerance. Another study showed that consumption of ashwagandha root extract capsule, called sustained released, or SR capsules, for 90 days, resulted in improved memory and focus, psychological well-being, and sleep quality and reduced stress levels. The study also showed that ashwagandha SR capsules were safe when administered over the period of 90 days. Effects against microbes. In traditional medicine, ashwagandha has proven effective against infections. Ashwagandha extracts have been proved effective against gram-positive Enterococcus species and Staphylococcus aureus, as well as gram-negative bacteria. The antimicrobial effects are achieved through several mechanisms, including but not limited to cytotoxicity, immune potentiation, and gene splicing.